Hello everyone, welcome to my channel Online Biology Tutorials. In this video, we will talk about some of the difficult problems that are a part of cbse Term 1 sample paper. Please be focused because I'll explain them logically to you. If you'll get the same type of question in your board Term 1 that is going to be scheduled in the month of November, you can do each question well. So the first question that I have picked for you is, how many types of gametes will be formed if the genotype of the parent is given? And here the genotype given is capital A, small a, capital B, capital B. Now remember, whenever we need to find gametes, we need to consider heterozygous pairs. Okay, so heterozygous pairs, they are only one in number, and that is capital A, small a. Capital B is homozygous. We will not be considering it. And the formula to calculate number of gametes is 2 to the power n, where n is the number of heterozygous pair. So simple, if you will put the value of n here, it will become 2 raised to the power 1. That means 2 gametes are there. Okay, so in this question, your option B will become the right answer. Let's take another example. Suppose you are being given this genotype. Capital A, small a, capital B, small b, capital C, small c, capital D, capital D. So in this, heterozygous pairs are how many in number? They are 3 in number, right? This one is 1, 2, 3. This one is not heterozygous because capital D, they are the same alleles and it defines the homozygous condition. So you will apply the same formula. It will become 2 raised to the power 3 and it will become ultimately 8 gametes. So any example, if it comes in the board Term 1 question paper like this, please follow the same logic. Next question which I have picked for you is, Rajesh and Mahesh have defective hemoglobin due to genetic disorders. Rajesh has too few globin molecules while Mahesh has incorrectly functioning globin molecules. Now before I come to see the options, one thing you have to remember, that thalassemia is a quantitative trait. And it is very much mentioned clearly in the NCERT, whereas sickle cell anemia is a qualitative trait. Means in thalassemia, the amount of hemoglobin will be less. This is the logic, whereas there would be a problem in the quality of hemoglobin in sickle cell anemia. So we have to retain this. And both they fall in the category of autosomal recessive disorders. Before coming to the options, these things should be there in our mind. Okay. Now Rajesh is written as too few globin molecules. So here I am writing Rajesh too few. Too few means they are talking about quantity. That means indirectly they are talking about thalassemia. Whereas Mahesh has incorrectly functioning globin molecules. That means indirectly they are talking about sickle cell anemia in the case of Mahesh. And both they fall in the category of autosomal recessive disorders. So this is the gist which we have created in our mind. Now we will look in for the options. First option says sickle cell anemia, autosomal ringed recessive trait. Second option says thalassemia and autosomal linked dominant trait. So dominant you will deduct in the beginning. So option A and D completely ruled out because they are talking about autosomal linked dominant trait. And neither of these disorders, they fall in the category of dominant trait. That means we have to look in between B and C. Our answer is already ready. Rajesh is suffering from thalassemia. I'm reading option B. And Mahesh is suffering from sickle cell anemia. So we already worked out, because we had initial knowledge, that thalassemia is a quantitative trait. So in the exam, don't waste your time. Just look in for autosomal dominant, because neither of these traits are autosomal dominant. You will deduct them and then you will work out like this if question is given, so that before coming to the options, your answer will be ready. This is the next question which I have picked for you. Here the transfer RNA structure is given and we just have to match site A with site B. Okay. Now one thing is sure here, that A site, this site of the transfer RNA, it is amino acid site. Okay. And the B site is the anticodon one, that would be containing anticodon. Okay. Now here, if you see, this is AUG in messenger RNA, and AUG always codes for methionine, right? So methionine is the amino acid, because amino acid is picked as per the codon on messenger RNA. So one thing is sure, that A will be methionine. So in this case, they have given you a table that has two columns, site A, site B. In this case, please rule out option A and rule out option D, because we have to pick up an option where amino acid will be mentioned, because it is not mentioned at A and D. So we are just ruling it out. We don't have to look at it. Now see here, another thing, messenger RNA chain is always 5 to 3 prime. Okay, and tRNA anticodon, it is always 3 to 5 prime. So here, if it is AUG, messenger RNA and transfer RNA, they are complementary to each other. So if it is A in AUG in messenger RNA, complementary base pair against A will be U, against U it will be A, against G it will be C. So for AUG in messenger RNA, it would be UAC in transfer RNA. And site A has already been worked, that this is methionine. So our answer will become methionine UAC. Okay, this is our right answer. So in your OMR sheet that will be provided in the exam, you will color option B for this question number. This is the next question, and this says concentration of which of the following substances will decrease. Decrease is the keyword here, in the maternal blood as it flows from embryo to placenta through umbilical cord. Now students, please remember, this is the embryo, okay, and this is the umbilical cord. This is placenta and this is maternal blood. Okay, this much connection should be learned by you. Now if the blood is flowing in this direction, means from placenta to umbilical cord to embryo, that means at this time oxygen will be more, amino acids will be more, because it is taking the nutrients from maternal blood to placenta to umbilical cord to embryo. Now in the question they are asking you which things they are going to get decrease. Now let's talk about carbon dioxide and urea also. When the blood would be returning back, because embryo is not using excretory system, so it is solely dependent upon diffusion. So CO2 and urea will be more as the blood will be passed from embryo to umbilical cord to placenta to maternal blood. So they are asking which concentrations will be at lesser level. So when they are going in, oxygen and amino acid, they are high. When they would be going back, their concentration would be less, because oxygen and amino acids are used by the growing embryo or the fetus which has become fetus, right? So embryo is the initial stage, fetus is the later stage where mini human being is formed. So answer to this is these two, option 1 and option 2. So in this case your right answer will become option A. So in Suppose this is question number 1. So along with question number 1, you will be given 4 options like this. Okay, there will be 4 circles. What you need to do is you need to color option A. You know, knowing answer is a strategy, but there are students who are making mistake in the OMR sheet. So carefully see the question number and color only one option. Okay, here in this case, picking up oxygen and amino acids means we will not tick 2 options. Rather, we have to move on and pick up where both the options are applicable. So be careful in such type of MCQs where they are asking for 2 options. This is very much important. This is our next question. To produce 400 seeds, the number of meiotic divisions required will be. Now to form one seed, you must know that one female gamete is required and one male gamete is required. So you know that when megaspore mother cell, it divides, we are talking about just one megaspore mother cell. When it divides by meiosis, 4 megaspores are formed, and out of which 3 degenerate on only one functional, and this one megaspore divides further by mitotic divisions to form embryo sac, and inside embryo sac only one egg is there. So overall logic is one meiosis is making one functional egg, whereas one meiosis in the microspore mother cell will form 4 pollen grains. That means one meiotic division will be resulting in 4 pollen grains, and all of them will be functional. So for 400 seeds, we would be requiring how many pollen grains? 400 pollen grains. So we require from one meiotic division 4 pollen grains are formed. So simple, we will take this value of 400 because our requirement is of 400 pollen grain, and we will divide it by 4. So that means we would be requiring how many meiotic divisions? We would be requiring 100 meiotic divisions to form 400 pollen grains. Now here we have already seen that one meiosis will result in one egg, this one, the circled one. Okay, so that means our calculation will be 100 for the male gamete and 400 meiotic divisions for forming 400 eggs. So we will just add on these values and we will get 500 meiotic divisions. Our answer will become option C. Now similar type of problems are recorded by me for chapter number 2. I will be sharing the link in the I button. Just go for that also. This is our next question, which is asking a cross is made between the tall pea plants having green pods and dwarf pea plants having yellow pods. In the F2 generation, out of 80 plants, how many are likely to be tall plants? Now see here, they are talking about the dihybrid cross. So let's make their genotype. It would be tall. For tall, we'll be taking capital T, small y, and they are saying that these are crossed together. So this is our dwarf and yellow, and here it is talking about tall and green. Don't get confused because this is just an example of dihybrid cross, but in your book, in your NCERT, they have given as homozygous dominant is crossed with homozygous recessive. Here they have not taken both the dominant characters together. Okay, so that's the only difference. We'll work out Here these are the gametes, okay, and this is our F1, capital T, small t, capital Y, small y. You know that when F1 is crossed with F1, we get F2. Now just come back. Now the same genotype is there as you received in the dihybrid cross that is given in NCERT. Now remember, in NCERT, in the F2 generation, phenotypic ratio was obtained as 9:3:3:1. Now, if I talk about the dominant traits, here they are asking out of 80 plants, how many are going to be the tall? And see here, tall is the dominant character, right? So, if you will see your NCERT, you will find at these two places where value 9 and 3 are given, at these two places that round character was given in your book. 9 were round and yellow and 3 were round and green. So round character was the dominant character. So it was exhibited, it was getting expressed at these two places, at these two phenotypic ratios. That means 9 plus 3. 9 plus 3 makes how many? That means 12 individuals were round in that pedigree that was given in the NCERT. So here we can consider tall as synonymous to round because we are talking about the dominant character. Now if you see how many individuals were formed in the dihybrid cross, 60, right? 9 plus 3 plus 3 plus 3 makes 16. In the Punnett square also 16 individuals were mentioned like this, right? So out of 12, fine, these are 12 individuals out of 16 individuals that are exhibiting dominant phenotype, and they have given us the value of how many plants? 80. So this is the way you have to do. 12 are exhibiting dominant character out of 16 individual formed, and we have to find out of 80 how many would be like exhibiting the dominant character. So this is very easy. You will do it mathematically and the answer will become 60. That means out of 80, 60 individuals will be exhibiting the dominant character. So these calculations, this expression is very, very important. Suppose if they will ask for any dwarf variety, similarly you can do for that as well. This is our next question where karyotype is being given to us. Karyotype is the diagrammatic representation of the chromosomes and we have to deduce the result. Now see here, all the chromosomes are being seen in the pairs. This one is the peculiar chromosome because at 21st position, how many chromosomes are seen? 3 are seen. That means trisomy is observed at 21st set of the chromosomes, and trisomy we have studied that this is seen in the case of Down syndrome. Another thing which we need to keep in mind is the sex chromosomes are similar in shape. So that means they are talking about 2 X chromosomes, and 2 X chromosomes means a female. That means the given karyotype is of the Down syndrome and that too of a female. Let's go for the options now. Affected, first is normal, that will not be there. Color blindness cannot be seen. This is a female with Turner syndrome, not possible. So we are there with the right option, and this is option C, the right answer. So they may give you any karyotype like this. They may give you of Klinefelter syndrome. In Klinefelter syndrome, you have to look in for the abnormal karyotype at the sex chromosomal position. Even for the Turner syndrome, every chromosome autosomes will be normal, but sex chromosomes will be abnormal. So only one chromosome will be seen in the case of Turner syndrome, and there will be no other chromosome, right? So the same genotype can be observed for Klinefelter. So you need to be vigilant at karyotype problems also. This is our next problem where they have asked for the labeling of the replication fork. Now, as per our previous knowledge, we know that this is strand is continuous, which is marked by 1. So we would be labeling like this. Labeling of the arms is very, very important. This will be our 3' end, this will be our 5', this will be 3', then 5', 3', then 5', 3', then 5'. Okay, so with these conclusions, we need to first make up our mind that 5 to 3' is a continuous strand. If it is continuous, that means it is a leading strand. Okay, now the first we have already got, so this is option first, and for fourth we have already find it out that it is 5 to 3'. For third we have already worked out it is 3'. For second we have already worked out that they are Okazaki fragments. These fragments are a part of lagging strand. Now let's see which option fit in, right? So 1 is showing discontinuous, it will be ruled out. Second, first is correct, that is fitting in. Second option shows discontinuous, of course. Discontinuous is applicable to Okazaki fragments because they are not stretched continuously. Third prime is 5', and fourth prime is this option is ruled out because these options are given wrong. Let's move on to option 3, which says C is 1 is discontinuous. So 1 we already worked out in the beginning. So that means we are left with this option. Let's see if it is fitting in. 1 is continuous. Yes. 2 is discontinuous. Yes. 3 is 3'. Yes. 4 is 5'. Yes. That means which is our right option? Option D. Now before I sum up with this, let's conclude once again. 5 to 3'. This is our 3 to 5'. Always remember the polarity that leading strand is always 5 to 3'. It will be continuous, where lagging strand will be discontinuous and it will show Okazaki fragments. Okay, this labeling is to be very, very thorough with you. Every year question is coming in and we are expecting this year too. This is our next question. It looks as if it is very, very difficult, but the conclusion is very easy. They are asking a stretch of a euchromatin has 200 nucleosomes. How many base pair will there be in the stretch and what would be the length of the typical euchromatin? Now you must know that one nucleosome, it includes 200 base pairs of DNA. How 200? 146 were included in the core plus 54 were included in the linker DNA. That means overall 200 base pairs were included, right? So they are saying that 200 nucleosomes are there and they are asking in the base pair. Very easy. 200 nucleosomes are given to you and you have to find the base pairs. So you will simply multiply 200 with 200 because one nucleosome has 200 base pairs. So 200 nucleosomes will have how many base pairs? 4 and 4 zeros base pairs. Okay, that means after solving this first part of this question, we can rule out option A and option B. That means we have to work out between option C and option D. Now let's find out what are they saying. They are saying what would be the length of the typical euchromatin. Now if you have studied the DNA structure, this is known as the pitch of DNA, which is 34 angstrom. Right? And 1 angstrom is equal to 10 to the power 9 meter. Now simply we will use the same value and we will multiply it with the base pairs. So it would be 34 into 10 to the power 9 and it will be multiplied with 4 and 4 zeros. Okay, so you will get your answer as 13,600 into 10 to the power minus m, because here we have converted this value to m. So in this case, option C will be our right answer. In the question, they may also ask you how many base pairs are there in just 200 nucleosomes. So you may get one question, one part of it also in the exam. The next question is the one which you have not done in the class earlier, but this is important. If Meselson and Stahl experiment is continued for 6 generations, because we generally talk in class for 2 to 3 generations, but here they have given a question on 6 generation, and they are asking that at the 6th generation what would be the ratio of these N15 by 15, hybrid and the light DNA. Now remember, for this I will be telling you the logic also, and I will be telling you a trick also, so stay tuned. These, suppose the red labeled are your N15 that we used in the beginning. Now our first generation started after it, so this further on it will be our first generation, and in this generation, if you remember, N14 or the lighter isotope was used, right? So I am going to show N14 with black color, and N15 I have already labeled with red. So in the first generation, you can see that we have received the hybrid, and the hybrid strands, they are 2 in number, right? Let's move on to second generation now. In this second generation, we have 2 double-stranded hybrids, right? So we are going to make them single-stranded. So this first strand will be separated like this, and the another hybrid strand will also separate like this. So as we are continuing now in N14, so the another strand will be N14 only. So in this you can see that hybrid, they are 2 in number, and N14 they are also 2 in number, as we are going in N14 only. Let's move on now to the third generation, right? So in the third generation, again same logic will be there. First we will be separating the strands. So our hybrid strand will be separating like this. Next strand, they are N14 only, and we will be separating them like this. Then again a hybrid that would be separating like one black and one red, and next one will be separating like this, right? So as we are going in N14 only, so next strand, the daughter strand will be N14 in nature. So I am drawing it with the black ink. So here we have how many hybrid? We have hybrid 2 in number, and we have N14 6 in number, right? So solving this type will be very much time taking for you. All you need to do is, you have understood the logic till generation 3. Now if you look in for first generation, this is our result. For second generation, this is our result. For third generation, this is our result, right? So if you see constantly, our hybrids, they are constant. That means they are only 2 in number. But see, N14 is continuously rising. So N14 in second generation, if you see in second generation, N14 strands were 2. In third generation, N14 strands were 6. Now I am telling you a trick how to find it out. Suppose you need to find it out for the fourth generation. How are you going to do? One thing is sure, that in the fourth generation, hybrid will again be 2. But what about N14? Now whatever number you have in third generation, it was 6. You will double it. So when you will double it, 6 plus 6, right? Plus 2 you will add on, because in the hybrid 2 strands are there. So it will become 14. So in fourth generation, N14 will be 14. Let's give one more example to you of the fifth generation. In the fifth generation, again hybrid will be 2 in number. And what about N14? We need to double 14 plus 14 plus 2 strands of the hybrid. So it will give you 30 strands of N14. Let's work out for 6th generation now. So in 6th generation, again hybrid will be 2 and N14 will be what it was in the previous generation, 30. So we will take 30 plus 30 plus 2. This means 62. That means in 6th generation, what is our result? We have hybrid how many? 2. We have N15 labeled 0. We have N14 labeled 62 strands. So this is our finding. Let's see what they have asked in the question now. The first value they are asking for N15, right? So this is 0. Hybrid they are asking, that is 2, right? This we have seen here. And N14 at 14 we have 62 strands. So if you will simplify, this ratio will become 0 is to 1 is to 31, right? And here is our answer. Option D. So this way, if you get similar type of question and they are asking for generation 3, 4, 8 or so on, please apply the logic. Do not solve it. All you need to learn is hybrid will always be 2. N15 will always be 0 and N14 will depend upon the generation. If you are moving from one generation to another, you just have to remember till generation 2. Hybrid 2, N14 2. If you want to calculate it for third generation, N14 will be 2 plus 2 plus 2. Because we have to double the value of 2 which is seen in the previous generation plus 2 in the end of the hybrid. So logically, please think you have to remember till generation 2 for next generations. Please don't make the strand because it will be very much time consuming. Hope it was interesting to learn. Stay tuned, and if you have not subscribed to my channel, please do share it with your family and friends. It is very much required. Wish you a happy learning. Thank you.